Assalamualaikum. So this video is for the paper 4 tips for biology paper. So let's go. We say this paper 6 also. So very first part, we're gonna talk about drawings. So if we are asked to draw a particular drawing, so how are we gonna draw? These are the rules that when there is like a drawing of diagrams of fruit, insects, or anything. So what are you gonna do? You're gonna follow these rules. That is to make sure use a sharp pencil. Your outlines must be clear. There should be no shading or sketchy lines. No arrow heads when labeling. And like straight lines, what do I mean by no arrowheads when labeling? So this is what I mean. Instead of drawing this arrow, you're gonna simply draw a straight line and label the diagram. And this is how you're gonna draw your diagram. There'll be no shading or no sketchy lines. And what is the word sketchy line means? Like these lines. Alright, so this is the step for drawing. If we go on and talk about magnification, which is the most appearing questions in exams, to calculate the magnification, so we're going to follow this particular pyramid for the magnification, which we have image size upon magnification and actual size. Alright, so this is how you're gonna solve your magnification questions, in which the length of animal A is 7.5 centimeters. So according to your ruler scale, so you're gonna convert the unit into millimeter by multiplying by 10, and the actual length is 135, and then we're gonna divide these both values to get the magnification. Now the next part is of comparison. Whenever we are given two animals to compare, what are we gonna do? These are the steps to follow. Make sure to compare the diagrams, the features which are visible in the diagram. If they are asked to compare the visible features, remember to observe the diagram completely and then write the comparisons, and use the labels on the diagram as your guide, and you can compare the number and shape, proportion, whatever. Yep, this is how you're gonna compare. Let's move on to design an experiment. Let's specifically talk about that you have to describe an investigation that you could carry out to show the need in seed germination for suitable temperature, the requirement of temperature. So means here temperature is an independent variable. We can change the temperature according to our requirement or investigation. So find the variable which is to be changed from the question, which is of course our independent variable. Here we have temperature. Then list all the variables you have to keep constant, which are known as constant variables, and then mention how long your experiment will last, and say how will you measure the experiment. You need apparatus and a set control for your experiment, how to minimize error by taking average values and means. So this is how we're going to answer a question in which we have written that two identical sets of apparatus are set up. Each set of apparatus should contain many seeds, all of the same species. Remember to take the seeds of same species and then placed on damp cotton wool in a glass jar, the lid of which is open to allow the entry of air. Of course, oxygen is necessary for the germination of seed. And then one set is placed on the bench in a warm laboratory, or better in a temperature controlled propagator. If you have to write this, you can write, otherwise it's optional. Then the second set is placed in a refrigerator at 4 degrees Celsius. Both sets of seeds are watered daily over a period of several days. Only the seeds in suitable temperature should germinate. Now to draw a graph, you have to use a sharp pencil. label both x's, choose appropriate scale of course, and the controlled variable which is time is plotted in x-axis and the dependent on y-axis, and then join the plotted points with ruled lines, means straight lines. And then in a bar chart or histogram, what is the difference between a bar chart and histogram? So let me clear. So basically this is your bar chart which is at a distance between these bars, and this is your histogram. So remember the difference between a bar chart and histogram. Alright. So this is the thing which I mean that you have to draw using ruled lines, means straight lines. Alright. Now here is the test for reducing sugars. For safety we need test tube holders, safety goggles, and lab coat, hair tied at back. If we have long hairs we need gloves, lab coat, right? And the procedure is mentioned right here. Pause the video if you wanna see. And this is the test for reducing sugars. Starch test is that crush the sample if it is needed or extract with water to make a solution, and then put the sample in a test tube, add iodine, drops of iodine, observe the color change. If it turns from yellow to blue black, it means there is starch present, and if it remains yellow means there is no starch. For protein test, add a measured volume of butyrate solution in a test tube or a beaker, and if the sample contains protein it will turn to purple or violet. So for the test of fats, add a measured volume of ethanol to the sample and an equal volume of water to the mixture. A white emulsion forms, which is a sign of the presence of fats. If no white emulsion, it simply means there is absence of fats. Now here are some tests for gases. For oxygen, carbon dioxide,